Good morning, guys. So today we are going to talk about meiosis. The main difference between meiosis and mitosis, it's pretty much that this is just going to make sexual cells instead of body cells, which is basically mitosis. So pretty much we are going to produce basically gametes that they are going to be the sperm and the egg cell. At the same time, they are going to be in the air reproductive of organs. In the case of the female, it's going to be the ovary, and in the case of the male, it's going to be the testicle. At the same time, we are going to talk about something which is that makes every single of us special, which is the DNA differentiation, which is going to be at the very first. We are going to talk about something which is haploid, which is when they get separated. So there's going to be a differentiation, and then they get together, which is going to be diploid. Once the two sexual cells they get united, this is going to be diploid, in which the resulting product it's going to be the zygote. So at this point, there's another human being in all this process. Now we are going to talk about the first stage, which is interface, in which the cell basically does the normal function of the cells, which is cellular respiration, and it starts to get prepared for the first phase, which is going to be prophase. So here we have prophase number one, which is going to be the first stage in which the chromosomes they are going to start to cross over, and they are going to start to share information between any single of them. So we're going to have a resulting information sharing. So here we have metaphase, which is stage number three. So in which we are going to have pretty much this information sharing, and they're going to basically we're going to have something. We got the homologs, and they're going to start to pair chromosomes and line up in the middle of it. Then we have anaphase, which is going to be the stage number four of it. So we are going to start to see like the spindle fibers begin to contact, and they are going to start to get pulled by the centrosome, and basically we are going to start to have this important thing that we are going to call basically the telophase, which is going to be the next part of it, in which they are going to start to get separated, and their cell membrane it's going to be pretty much two new different cells. So after the first cytokinesis, we go back to the prophase. So in which this one it's going to be the same. They are going to start to cross over sharing information, and pretty much do the same process. They are going to start to move to the poles, and there's going to be a cell membrane. There's going to be a nucleus, in which all of them they are going to be pretty much two different cells, but they are going to be obviously the same type of cell, which is going to be gametes. So right here we go back to metaphase, in which the spindle has formed completely, and we are going to see like they are going to start to line up in the middle, and they are going to get ready for the next stage, which is going to be pretty much or beautiful anaphase, in which they're going to start to get pulled over by each centrosome, and they're going to create basically the next phase, which is going to be a beautiful telophase. But pretty much at this time they are just getting ready, and they are getting separated. So the sister chromatid gets separated. They are pretty much the same. So right here in telophase and cytokinesis, they are going to start pretty much the cell membrane. It's going to start to get divided, and we are going to have at the very end after cytokinesis, which is going to be when they are in inner phase, and we're going to have four different type of cells, which is going to be the same. We're going to have, in this case, we're going to have the male cell, which is going to be sperm. We're going to have foreign sperm at the very end. So there's something interesting right here. As you can see, they are not the same. So for example, we are going to just answer just brief questions at the very end. So for example, what would be the four final cells look like if the crossing over didn't occur? Basically, they are going to look the same, which is dangerous because we need that cell differentiation in order to don't get killed by the same bacteria or virus. So that's important. So the next question is gonna be, why is crossing over and independent assortment important for the genetic difference? Basically, because first of all, the crossing over is going to start to share information between all the chromosomes, and then after that, the independent assortment, which is pretty much the when the two cell membranes they get divided. This is going to give us like different cells, and this is going to give us genetic diversity because we need it in order to survive to bacteria and all that we have been surviving as a group. So sometimes no disjunction can occur, and the chromosomes don't look separate properly. If that happens, what will your final four cells look like? So basically, there's not gonna be like four cells. There are gonna be pretty much like two, but it's so rare, and this is going to bring us like malfunction and the non-creation of a new human being, basically. I hope you enjoyed the video, and just always remember to stay curious, and just share any type of information with your partner in order to learn as much as possible in your life. Have a good day. See you later.